This video focuses on Krabbe disease. We will go over the background of the disease, general information about it, its pathogenic mechanisms, and advances in treatments and new discoveries. The common name of this disease is Krabbe disease, named after Danish neurologist Knud Haraldsson Krabbe, the first person to describe the disease in 1916. This is a rare disease affecting approximately 1 in 100,000 live births in Europe and 1 in 250,000 births in the United States. This disease is also referred to as globoid cell leukodystrophy. Globoid cells are characteristic of the disease. If somebody is observed to have these cells, they have Krabbe disease. They are large and abnormal, containing multiple nuclei. Leukodystrophy is a group of rare genetic disorders that affect the brain and spinal cord's white matter, the tissue made up of nerve fibers, or axons, covered and protected by myelin. 90% of Krabbe disease symptoms begin in individuals between 1 and 7 months of age. This is classified as the infantile onset form of the disease, and it is rapidly progressive, with a life expectancy of only 2 to 3 years. The remaining 10% of cases are classified as either the late infantile, juvenile, or adult form. These forms progress more slowly the older the age of onset is. Life expectancy varies, but it's usually about 10 years after the onset of symptoms. Typically, in the most common infantile form, there is regular growth and development until 4 to 6 months, followed by restlessness, muscle weakness, hypersensitivity, vision and hearing loss, abnormal posture, and slowed mental and physical development that worsen. Symptoms of later forms of Krabbe disease include abnormal gait, irritability, seizures and tremors, blindness, deafness, impaired movement control, burning sensations in the limbs, and overall mental and physical deterioration. This severe neurological deterioration, loss of bodily function, and varying complications are eventually fatal in all types. How does one get Krabbe disease? This disease is a result of a genetic mutation. More specifically, it is an autosomal recessive disease. Autosomal meaning the mutation is not on a sex chromosome but a numbered chromosome, chromosome 14 in this case, and recessive meaning 2 copies of the mutated gene, one from each parent, must be inherited for the disease to be expressed. The mutation affects the galactocerebrosidase, or GALC gene, which is found in cells that compose the nervous system. When this gene is mutated, the cell does not have the instructions to make the functional enzyme. In normal, healthy cells, the GALC enzyme is responsible for breaking down certain sphingolipids. This is a class of lipids that make up cell membranes, and this is for lifelong maintenance. The particular sphingolipids are galactosylceramide, or GAL-CER, which is a component of myelin, and galactosylsphingosine, also known as psychosine, which is a toxic byproduct of myelin synthesis. Breaking down these sphingolipids, particularly psychosine, is very important since if no breakdown occurs, it will accumulate and it is neurotoxic. The accumulation of psychosine seen in the absence of the GALC enzyme in Krabbe disease is particularly harmful to oligodendrocytes and Schwann cells, both crucial to nervous system function. Oligodendrocytes wrap around axons and produce myelin sheaths, the structure that protects and insulates our nerve fibers, and Schwann cells also wrap around axons producing myelin sheaths, as well as regenerate damaged axons. Oligodendrocytes do this in the central nervous system, and Schwann cells in the peripheral nervous system. If psychosine accumulates, it integrates into these cells' lipid bilayer, which disrupts the membrane integrity. This causes impaired cellular signaling, mitochondrial dysfunction, and apoptosis. The deterioration and destruction of these cells causes demyelination, and these events trigger an immune response from the body that involves the activation of microglia, the brain's immune cells. The microglia arrive on site, but upon being exposed to the accumulated psychosine, some of them get converted into globoid cells. In their attempts to alleviate the cell damage and destruction, the microglia cause inflammation, and the globoid cells worsen it, which worsens the overall disease progression and symptoms. To simplify and sum up all of those mechanisms, the lack of the GALC enzyme in Krabbe disease causes psychosine accumulation, which is toxic, causing damage and death to oligodendrocytes and Schwann cells. This causes demyelination in the central and peripheral nervous systems and severely disrupts neural communication. This contributes to the motor, sensory, cognitive, and behavioral symptoms that are associated with Krabbe disease. Neural networks experience a loss of connectivity, and these networks are crucial for mental and physical development and capabilities. The neuroinflammation from microglia and globoid cells further contributes to the demyelination and death of cells in the brain and these symptoms. The lack of protective myelin makes axons vulnerable to damage, which is another contributor. A biochemical pathway that is affected by Krabbe disease is the sphingolipid metabolism pathway. As mentioned, sphingolipids are a class of lipids that compose cell membranes. This pathway has many functions, including cell proliferation and survival, apoptosis, immune regulation, and inflammation. This is a large, branching pathway, and the main points are summarized here. The molecules in brown are responsible for varying protective, stabilizing, regulatory, and communication roles. The portion of it we're interested in and have already discussed involves galcer and cycosine, the sphingolipids that do not get properly broken down in Krabbe disease because of the lacking GALC enzyme. As we now know, this is extremely problematic and is the root of Krabbe disease. It's also worth pointing out that while this shows galcer's conversion to cycosine, the effects are not significant as it occurs to less than 1% of it. Unfortunately, there is no cure for Krabbe disease. However, there are treatment and management options that help slow the progression of the disease to prolong individuals' life expectancy. Hematopoietic stem cell transplants, or blood cell transplants, are good options since these donor stem cells differentiate into microglia cells. This populates the brain with cells that have the functional GALC enzyme. Blood cells in the umbilical cord have the GALC gene and are often used. To manage symptoms, there are various recommendations, including using muscle relaxants to limit spasms, seeing a physiotherapist to increase mobility, an occupational therapist to improve motor function for children, a speech therapist to maintain communication abilities, as well as using feeding tubes if the muscles involved in swallowing are weakened. A study done in September of 2023 made an interesting new remark about Krabbe disease that may inspire new treatment options and hopefully bring the field closer to developing a cure. Researchers observed high levels of CD8 T immune cells in brain and spinal cord tissues in a mouse model of Krabbe disease. The presence here of these cells was 9 to 10-fold higher than in normal healthy tissue. CD8 T cells are also known as cytotoxic T cells, and simply put, they kill cells that have been infected with bacteria or viruses, as well as destroy tumor cells. In this study, researchers determined that the CD8 T cells largely contributed to the harmful neuroinflammation that is seen in Krabbe disease as a part of the immune response. Selective blocking of their function using antibodies preserves the integrity of neural tissue and reduces inflammation. This would translate to a decrease in the severity of motor, sensory, cognitive, and behavioral symptoms. These findings suggest that CD8 T cells may drive this disease and its progression, and therefore targeting these cells could be a possible therapeutic approach that should be investigated. The potential of this approach could also be investigated in similar diseases.